Next week, we have what I think is another extraordinary program on the great neglected diseases. And you might think about it. We talk about, many diseases here which have huge numbers of people involved, HIV and several others, tuberculosis. But there are diseases, quite a few, that affect millions and millions and millions and millions all around the world, and they sort of come under the radar. Most lay people don't know anything about them, and I suspect most scientists here may not be aware of the great neglected diseases. And the fact that they're neglected means there's a tremendous opportunity for imaginative research. It's not that there aren't good, intelligent, creative people working on these diseases, but that it desperately needs the kind of bridging, such as illustrated in our logo. For those of you who don't know, that's the Brooklyn Bridge, and you are, like the gentlemen who are on the catwalk, linking an exciting area of basic science that's the Brooklyn side, with an enormous impact on human animal physiology and health. We'll get to that. The following week after that, I would mention to you that we really have an extraordinary opportunity. John Mather, who is the director of the Webb Telescope project at NASA, and was the lead investigator, received a Nobel Prize for the Kobe study of the formation of the universe, the Big Bang analysis. So he's going to talk about cosmic imaging. And Jennifer Lippincott-Schwartz, who I suspect is known to all of you, one of the great leaders in dynamic imaging of cell biology. Jennifer's going to talk about imaging at the molecular level, a different kind of bridge. It should be an interesting thing. So I just want to say a couple of words. So this is the basis for the discovery of the innate immune system. The discoverer, Metchnikov, Eli Metchnikov, was a biologist, and he was studying a whole variety of living things with a microscope of the day. This is mid-19th century, so simple microscope. And what he observed was when they wounded the larvae of a starfish, he could see cells that were coming in to repair the wound. And he said these were like resting, wandering cells, which now we call macrophages. And he believed that they were the basis of the response to injury in any species. That was the nature of his work. He shared the Nobel Prize with Paul Ehrlich in 1908 for this. Now, typical in science, and I bring this up because it's very relevant today, we do research which goes from A to B, and that's the highway. But of course, we've all learned that there's very little A to B in biological sciences. There are too many C's, D's, E's, F, and the whole thing is kind of a network. But we believe in our A to B, and somebody else believes in their C to D, and you need a bridge to get people to talk together, and this is a good example. So Metchnikov discovered phagocytosis, and he said that is the basis by which any species responds to injury. It is the innate immune system. It's with us. Now, there were other great, and in those days even greater, individuals by reputation: Pasteur, Behring, and a whole group of their fellows and so forth who disagreed, and they insisted that serum antibody-mediated immunity was the key. That was the way. Forget about this innate system. And of course, they would immunize an animal to, say, diphtheria toxin and then expose them to diphtheria, and they didn't get diphtheria. So this was a whole explosive area which ultimately led to the discovery of T cells and B cells and so forth. And of course, it turned out that both of these groups, which fought each other tooth and nail, and Metchnikov lost. People forgot about him for a long time. But it turns out, of course, that both are correct. And his work was neglected until recent times. He's kind of been resurrected and is sort of widely recognized as the father of immunity. But there's much more to Metchnikov. Being a biologist, he wasn't just interested in the starfish thing. He was interested in life, and he wanted to live a long time. And so he got involved in the study of aging in the early 20th century. And I don't really know, maybe Yasmine can inform us, how he actually got into the fact that the gut bacteria were like a separate population in our body and that they were really important in health and disease. And he found that, believed that if he could replace those bacteria with lactic acid forming ones, the one that he did most of his work with was a Lactobacillus bulgaricus, which is widely used in yogurt. He was impressed that there were a lot of very old people who were old, he thought, because they'd been eating yogurt. And when he died at about the age of 65, I think, his biggest distress was that his opponents would say, see what that yogurt did for you. You didn't make it beyond 65. But he had another brilliant idea. Do some bacteria produce disease? These are gut bacteria, through producing toxins. And he developed this idea of auto-intoxication, which is today's world. And it led to the idea of whether you can replace the gut bacteria and prevent disease. And he speculated that maybe it could even delay aging. This was sort of the first probiotic. He gets a bad rap because he never said you could delay aging. That was a real hypothesis, and I think some newspaper person took advantage of it, and he became known as you eat yogurt and you live to be 90. At any rate, he was way ahead of the pack. And we're very delighted to have two colleagues at NIH who are also very much involved, and I would say right up there with the pack. Our first speaker is Yasmine Belkaid, who, I should have mentioned, Metchnikov ended his career by being the director of the Pasteur Institute, which is where Yasmine got her Ph.D., sort of like handing the torch. A few years in between, but what's the difference? And there she studied leishmania and immune responses in the innate system related to leishmania. After that, she spent some time in the Children's Medical Research Foundation in Cincinnati, where she continued her work on the innate immune system and in relationship to the gut bacteria. And along the line began to study germ-free animals. And this was a major breakthrough and has led to extraordinary findings. She came to the NIH and has continued her outstanding research. Her honors and so forth would keep us here for a long time. But I would mention she was elected to the National Academy of Science, and I think it was last year received the Sanofi Pasteur Award for the excellence of her science. And we're very delighted that she's with us today and is going to sort of tell us where we stand Now, the second speaker is Barbara Rehermann. Barbara is the chief and senior investigator in the immunology section of the liver disease branch in NIDDK. She graduated in medicine in Germany, was interested in immunology, and spent a very productive postdoctoral fellowship with Francis Cesari at the Scripps Institute. Cesari has really been one of the leaders in the development of understanding of the immunology related to viral infection of the liver. And that is an area in which he has continued to work. Now, in addition, she had an extraordinary paper that was published in Cell a couple of months ago, which I'm sure many people, I enjoyed reading immensely, and I usually can't get through many papers written in Cell. But what she discovered was the microbiome differences between wild mice from Maryland and standard mouse strains, and what happens when you change these in relationship to immune responses. So enough of introduction. It's a great pleasure to have you both here and look forward to your talk. Yasmine.
So thank you very much for being here. Thank you very much for inviting me to present. I don't think I'm going to tell us where we stand because I have absolutely no idea. What I'm going to just try to do is actually give a very big overview of some of the main understanding of the relationship in the microbiota and essentially the immune system and the relationship with pathogens. So, of course, this is a mandatory slides that most of you have seen numerous times. Once again, we are a composite of cells, human cells, and microbes that are not only bacteria, of course, which is what has received the most attention over the last few years, but also viruses, fungi, protozoan, and so forth. These microbes colonize all body surfaces very soon after birth. There is actually a very important role of this microbe early in life in the development and seeding of the immune system. These microbes are, of course, highly diverse, and they are believed to be at least as numerous as the number of cells in the human body. And the number of genes expressed is very likely higher than the gene expressed by our own immune system. So, of course, it's not a new finding that we are colonized by microbes, and quite a long time ago now, someone that has a name that I cannot pronounce, I was able to actually show via his discovery of the first primitive microscope that we were colonized by microbes. And I think these images actually are what he drew when he observed, I think, his saliva. And basically, the uncovering that actually we are colonized constitutively by microbes was really done quite a long time ago. Very much in line with what Irvin was actually mentioning, of course, Metchnikov actually uncovered an association between microbe and potentially beneficial effect for the immune system and the health of the individual. But as it was mentioned earlier, he got it a bit wrong. What he actually believed is very much then the microbiota was actually toxic, and you needed really to replace it by the beneficial microbe. He saw the microbiota as his potential enemy. And I'm going to discuss with you the fact that it's actually there is some truth in it, but very clearly this microbe also play a very fundamental role in positive elements in both the immune system but also physiological system of the host. What was really an interesting observation was done actually by a German scientist that really uncovered for the first time the association between lactobacilli and the health of the vaginal ecosystem. And he was really the first one to show this colonization resistance, which is the fact that a certain class of microbe, when present in the tissue, can prevent colonization against pathogens. This was really a very fundamental discovery that today actually is the base of a large number of exploration and also therapeutic approaches. So in very, very large overview summary of now maybe something like 15 years of work, the microbiota has been linked directly and indirectly to a numerous aspect of host physiology. So of course, this microbe play a fundamental role. I'm going to try to see if I can find, fundamental role, of course, in nutrition and absorption, as well as vitamin synthesis. They are, of course, this fundamental element in the GI tract. However, the microbes that live within the GI tract are also very important to also control the metabolism of the host, and really some fascinating work that was done early on by Jeff Gordon by transplanting microbiota from obese animals to actually lean animals, or obese individuals to lean animals, was able to transfer metabolic syndrome, which is quite remarkable. there is actually an emerging line of evidence linking the microbiota to behavior and cognitive function, and in particular, alteration of the encounter of certain microbe early in life can potentially lead to cognitive function disorders, at least experimentally. But importantly, these microbes do not only control the local immune system in the GI tract or local function of the GI tract, they also have a fundamental role in the control of the whole immune system of the host and the whole physiology of the host. So they act locally and systemically. And in particular, I'm going to discuss with you today is essentially the relationship of this microbe with the immune system. So work that was done by many groups, including ours, over the last few years, highlighted the fundamental role of these microbes in controlling the immune system, which means that these microbes actually play a role in different aspects. First, they can actually lead to the development of the immune system. Certain lymphoid structures, for example, will need the action of the microbiota to be properly developed. The microbiota can also influence the quality and the number of hematopoietic cells development. They can actually act in the bone marrow by controlling hematopoiesis. The microbiota can also control the immune function. It has actually been shown, I'm going to discuss some of those mechanisms later, then it actually allows the induction of immune responses to pathogen, both B cells and T cell responses, and protection of the response to pathogen. And the microbiota, of course, can play an important role, what is called the functional tuning, which means that the microbes that live in the tissue are able to induce the production of local factors that have the capacity to activate directly cells that enter in the tissue. And it's very important for immune cells when they enter in the tissue to receive these terminal signals that allow them to produce cytokines and other mediators that are important for the control of infection, for example. Because of this fundamental role, of course, a large array of work has actually shown that the microbiota can help to control infection and really represent an important adjuvant of the immune system. There is evidence, and in particular for all vaccine, that the microbiota can actually promote response to vaccine. And importantly, really an exploding area of research right now, the microbiota has been linked to the success of immunotherapy. Really, the first line of research linking the microbiota to the control of cancer was done within the NIH by a group of Giorgio Trinchieri and Romina Goldsmith that actually uncovered an important role for this microbiota in promoting the control of tumors. And more recently, this was actually developed and further developed by other groups in the context of immune checkpoint therapy. That is, of course, a true revolutionary approach in the context of cancer. So really a large amount of research today is to try to understand the mechanism by which this microbe are actually able to boost the response to cancer and how these differences can potentially explain some differences in response to treatment. So of course, because they control the immune system, these microbes have also been linked to a large number of inflammatory disorders. There is actually evidence that the microbiota can actually control the development and amplitude of autoimmune responses. As I mentioned before, via the control of the immune system and other factors, the microbiota can also control metabolic syndrome and development of these disorders. And there is actually very nice work highlighting the microbiota, and in particular early life encounter of certain microbes with the development of atopy. There's a very nice study that was done by Brett Finlay recently in Canada, looking at children that receive antibiotic treatment, was able to show that actually early life exposure to antibiotic really very much is associated with the enhancement development of atopy. So really a nice study in human settings linking the microbiota to the emergence of inflammatory disorders, that is the wrong microbiota. So one of our research that we have spent a lot of time exploring is really the crosstalk between the microbiota and pathogens. And the reason we were very interested by this question is, of course, the fact that all those tissues that are really the primary site that are targeted by pathogen, the skin, the lung, the GI tract, and so forth, are also the natural habitats of the microbiota, which means that the first encounter with the microbes that infect an individual with the host is really in this milieu that is naturally colonized by these microbes. So really, the number of interactions are quite complex, and this is really important to understand this relationship between this form of organism. When we need to think about the microbiota, coming back to Metchnikov actually theory, it is true that the microbiota is also a reservoir of pathogen. The microbiota of any of us at any given time contain a large number of microbes that can, in a contextual manner, cause a disease. Part of this microbe, in some cases, can be antibiotic-resistant microbes. And these actually microbes, under normal circumstances, are held in check within the GI tract of a person or the skin of a person because of the action of the microbiota against them, and I'm going to come to this point later, but also the action of the microbiota on the immune system that prevent the colonization by these microbes. So it is important to remember that the microbiota is also naturally colonized and actually contain microbes that can contextually cause disease. So work that was done by many different investigators, coming back a couple of centuries ago, really highlighted the importance of this microbiota as a first shield against infection. So one of the primary mode of action is actually indirect, and it's going to be via the induction of the innate immune system. Beautiful work that was done by Laura Hooper, for example, highlighted how the gut microbiota can actually influence the production of certain antimicrobial peptide that can limit the outgrowth of certain microbes. As we and others have shown, of course, this microbe can also promote adaptive immunity that in turn can limit the development of this infection. There is also evidence that metabolic product of the microbiota can also have an effect on the ability of this microbe to colonize or actually to just become infectious. And finally, there is also direct evidence of the action of the microbiota. One of the fundamental one is, of course, competition for resources. Because these microbes are there and consume resources, they can also prevent colonization by pathogen via the sheer competition for the limiting amount of nutrients that are present. And this kind of phenomenon may be very important, for example, in the skin, in which the nutrient is actually very low. And there is, of course, direct toxicity, and these microbes that are present in our own GI tract or skin can produce molecules that can themselves actually can have an antimicrobial effect. So via all this different activity, the microbiota is really able to provide this fundamental shield that prevent colonization with a large number of pathogens. So an example of this fundamental relationship can really be exemplified in the context of antibiotic treatment. And in some patients that are treated with multiple antibiotics in clinical settings, for example, this patient can unfortunately be monocolonized by an antibiotic-resistant pathogen that can really take over the entire GI tract, for example. And this can really lead to very dramatic consequences. So this actually led different scientists, including Eric Pamers and others, to develop approaches that try to actually prevent this exuberant growth of microbes via the utilization of microbial cocktail that can, in a very rational way, compete with these microbes. And importantly, this has also led to a true revolution in the treatment of C. difficile recently. That is the best example of how we can utilize microbiota transfer to control disease. In 97% of the case, patients that are really colonized by C. difficile, that is, of course, a life-threatening chronic disease, can actually be cured from this infection just by the transfer of the microbiota of healthy individuals. So really, in some cases, utilizing microbiota transfer can have a profound consequences on the ability to control infection. And this approach may be very important for the control of antibiotic-resistant pathogen, for which we really have very little resource today to actually control. So as I mentioned before, this approach now is actually in development to try to actually control other antibiotic-resistant pathogens. And importantly, the creation of those optimal cocktails is actually now the next phase of this field of microbiota transfer, to try to not only transfer the whole microbiota of individuals, but understand what will be the best cocktail of microbe to transfer to control infection. This approach is actually currently also developed in the context of cancer therapy to try to see if actually defined cocktail can actually boost response to immunotherapy. So as I mentioned before, the microbiota plays a very important role in the control of immunity to pathogens. And some of this ability of controlling the pathogen is via the ability to promote cytokines or factors that in turn have the ability to reinforce tissue and barrier responses. So our own journey into the crosstalk between the microbiota and the pathogen led to complex studies that actually led to understanding that actually virtually every step of host-pathogen interaction can be controlled directly or indirectly by the microbiota. For instance, yeah, something strange happened there, but transmission anyway. So the transmission of the pathogen can actually be in some cases controlled by the microbiota. I'm going to explain later what I mean by that. Importantly, the microbiota can control immunity to pathogen via its ability to promote T cells and B cell responses. The microbiota can in some cases control pathology. I'm going to give you also an example of this phenomenon. And finally, the microbiota can in some cases promote tissue repair. So our work in the exploration of the crosstalk in the microbiota and immune system started quite a few years ago by a very talented team in the laboratory, Nicolas Bouladou and Jason Hall, who actually did a very simple experiment, which is actually to treat animals with oral antibiotic and look at the ability of these animals to control an infection. And what they were able to show is if actually you treat the animal with antibiotic, the ability of the cells to produce in the gut IL-17 or interferon gamma was actually significantly decreased. And actually, this phenomenon was linked to an enhanced ability of the microbe, in this context, Microspordia, to colonize the mice. So this line of work really led to an understanding that actually the gut microbiota could actually play an important role as an adjuvant of the immune system. Some of you may be also very familiar with the work of Dan Littman, who actually uncovered very nice association between defined microbe and in particular SFB, that is a microbe that can be found in mice, and its ability to promote very specifically Th17 responses in the gut. And via this ability, he was able to do a very famous experiment today that is actually to utilize mice from different facilities or using microbiota from different mice, and was able to show that if the animal contain SFB or do not contain SFB, they are vastly different in the ability to control Citrobacter rodentium in this specific example. So again, this was an example that a defined class of microbe could actually boost the immune response in a very specific way. So this was actually subsequently shown in a large number of diseases, some of them infecting the gut, some of them actually affecting the lung. For example, influenza has been shown experimentally to be controlled by the quality of the microbe that live in the GI tract. And this was also shown in the context of Salmonella, Plasmodium, and many other pathogens. So what we need to remember is, of course, as I mentioned before, the microbiota is not just bacteria. And there is actually nice evidence that the ability of this microbe to boost the immune system is not just due to bacteria, but can also be due in some cases to protists. And some work that was done a couple of years ago now by the work of Mirad showed that actually in animals that are colonized naturally by a protozoa, in this context Trichomonas musculus, the animals are actually better able to control an infection to Salmonella, but also were also better able to control some class of tumors. So this actually really shows that in certain cases, protists may be sufficient or able to push the immune system. Work that was done also recently by Ken Cadwell at NYU showed that actually certain virus can actually be sufficient in some cases to push the immune system. So this actually clearly shows that many different microbes may have the ability to engage the immune system, and the cooperative action of all those different microbes will be actually what is required for the optimal control of the immune system. But what we need to think about the microbiota, we need to think beyond the bacteria to also other forms of organisms that are naturally present in the different tissue. So I wanted to actually discuss with you a very important point about the microbiota in terms of its negative role in some cases. So it has actually been shown in the GI tract in particular that certain class of infection and pathogen can actually be promoted by the action of the microbiota. This was actually first shown in the context of worm infection. It was a work of Richard Francis that showed that in the context of Trichuris muris, the eggs that actually arrive in the GI tract needs the encounter with the microbiota to be able to hatch. So for this pathogen to actually be able to infect the animal, it needs to encounter the microbiota, and in particular canonical molecules produced by the microbiota. Work that was done by Julie Pfeiffer highlighted the importance of the gut microbiota, for example, in the transfer of certain viral infection, especially oral viral infection, that require the action of the microbiota for the virus to be infectious. So in some cases, infection itself can actually be promoted by the action of the microbiota. As I mentioned before, in certain cases, the microbiota can also be the cause of the pathological response. And I'm going to just explain what I mean by that. So work that was done by Laura Hooper again a few years ago really nicely really highlighted this importance of the separation between the epithelial structures that is actually in the gut, that is actually visualized here in blue, and the bacteria themselves. So in most cases, especially in the GI tract, there is really a separation between the tissue, which is actually the gut here, and the microbe. This is really due to the cumulative action of the mucus, of course, that is a very important structure, antimicrobial peptide, and antibody. All those together really push these microbes to make sure that we can actually sense them, but in a way that is very controlled. So what infection does is actually changing this relationship quite dramatically. And in particular, in the context of an oral acute infection, what we had found a few years ago is that the separation between the microbe and the immune system is no longer seen. And you can see here that you have no longer any separation between the epithelium and the microbe, and then these microbes are really clearly able to invade the tissue. More than that, in the context of an acute infection in some cases, the microbe escape the gut and can actually be grown in the periphery. And this massive translocation of microbial product has actually been reported in other systems in context of experimental model, but also in the context of HIV, in which this microbial translocation of product or bacteria has been now proposed as a mechanism to enhance immune activation and therefore the propagation of the virus. So this phenomenon is one of the associations that happened with the microbiota and the pathogen in the context of an inflammatory responses. Another thing that happened in the context of infection that can really threaten our relationship with microbes is that the composition of the microbiota can change dramatically. This certain class of microbe can thrive on a product that derive from the inflammatory process, and this will be the case, for example, of gamma proteobacteria that become very dominant in the context of an infection. In the context of acute infection, in most cases, you have a dominance of the GI tract by E. coli, for example, that in turn become responsible of the pathological response. So in some infection, the pathology is not due to the pathogen, but due to an aberrant growth of certain microbe that in turn can cause disease. So in some case, the microbiota can really be a causative element in the pathogenic process. So clearly, there is a profound association between the microbiota and pathogen and the immune system. But what actually is less clear is really the mechanism utilized by this microbe to engage the immune system. And I'm going to just spend the next few minutes of my talk to highlight some of the examples of the mechanism utilized by this microbe and how different this class of immunity we mount to commensals could be from an immunity to develop against a pathogen. So most of what we know about how the immune system works comes from the work we have done as immunologists, exploring the encounter of pathogen with the immune system, which is in the context of aggression, in the context of tissue damage, inflammation. But we really understand very little about how the immune system communicates with a microbe that is not an invader, a microbe that is not causing disease. So the class of immunity that is developed, the quality of this response, the antigen that are seen remain vastly unknown. And this is something that we have actually over the last few years decided to begin to explore. And the tissue we have actually decided to focus on over the last couple of years to address this specific question is the skin. The skin, as all the other body surfaces, is colonized by its own microbiota. And work that was done by Julie Segre, Heidi Kong, and many others have highlighted the complexity of this microbe in the skin microenvironment. These microbes are present for certain on the surface, but more importantly within structures in the skin. They will be in hair follicle, sebaceous glands, sweat glands. And in this tissue, at the difference of the GI tract, the biomass of this microbe is very low. And we actually decided to explore this tissue in part because of this reason, because looking at mechanistic insight may be simpler in a system that is actually less rich in microbes, in which actually certain microbes may have a more dominant effect without a large number of others. So work that was done quite a few years ago now in the laboratory by a very talented graduate student, Rudy Naik, who started her laboratory recently, uncover an important role for the skin microbiota in controlling the skin immune system. And she was able to show that this ability of the microbe to control the skin immune system was at steady state independent of the gut microbiota. So that's actually able to show that each community of microbe living in different environments may be able to control the immune system locally. So over the last few years, we actually have developed quite a few collaborations with people to just have access to primary isolates coming from healthy donors, but also from animals, some of them actually coming from Barbara and others, to really look at the ability of different microbes to engage the immune system. And the goal we have is really to try to see if we can uncover unique interaction between different classes of microbes, and actually using this interaction as the mode of exploring the mechanism that is used by the immune system and the microbe to communicate. So the approach we use is simple. We actually take microbes, we apply them at the surface of the skin, even at a concentration as low as what is actually believed to be the human density of microbe in the skin. And we look at the ability of this microbe to engage different class of immune cells, allowing us to ascribe a very specific immune signature to different class of microbes. And this actually collectively allowed the laboratory to uncover association between different classes of microbe and the immune system. And the point that I wanted to make with this slide, very much in line with the work that was done by Kenya Honda and others in the GI tract, is in many cases, microbe can have a very specific ability to engage the immune system. So it's actually not rare to find microbe with a preferential ability to engage one subset of cells versus another's. And there is some extreme case, for example, in which Corynebacterium, for example, in the skin is really able to exclusively activate gamma delta T cells and no other population of cells. So this actually association between defined microbe and immune system and immune cells give us now an opportunity to look for mechanism. And I'm going to just briefly discuss some of the work that was done in the laboratory by Shruti, but also John Linham, that actually has done most of the work on the last part that I'm going to mention. So a few years ago, we had actually found that if you apply the skin of an animal with actually a bacteria, and in this case, this is not germ-free mice. These are mice that contain their own microbiota. If you apply the animal with Staph epi, you can actually lead to the accumulation of CD8 T cells in the skin. The CD8 T cells are intriguing. They produce some 17, some gamma. And this effect was really restricted to Staph epi and not other population of microbes. So Staphylococcus epidermidis is a very common microbe. All of us have it at any given time. So it's a very common skin microbes. Work that was actually done by Alison Bird, actually looking at different isolates we obtained from the group of Julie Segre that were fully annotated, was able to show that this ability to activate the CD8 responses was actually restricted to a very specific clade of bacteria. and not the others. And that's actually make an important point. That is, when you want to understand the association between the microbe and the immune system, looking at just the species will never be sufficient, and you may really have to go to the clade level, and in some cases, really to the isolate level. So there is, in some cases, an extreme level of sophistication in the ability of unique microbe to engage the immune system. So to summarize some of the findings what we had in using this phenomenon, we're able to show that those CD8 T cells that are generated in response to this encounter with this microbe are able to migrate to the skin. They do not cause any inflammation, which is really a striking feature of response to a commensal as opposed to a pathogen. Those CD8 T cells can migrate to the keratinocytes layers where they can actually promote antimicrobial peptide production. And this actually has for consequences to block the ability of Candida, in this case, to actually infect the animals. So this actually showed that adaptive response to a commensal that happen in a very quiet manner, non-inflammatory manner, lead to generation of cells that can go to the epidermis and actually promote antimicrobial defense against pathogens. So really creating a heterologous protection against subsequent infection. So more recently, work that was done by John in laboratory was able to actually show and compare cells that are generated against a pathogen versus cells that are generated against a commensal. And for that, he purified CD8 T cells generated against HSV versus CD8 T cells generated against Staph epi and compared them, and was able to show by looking at gene expression, and very surprisingly, the cells generated against the commensals have a very unusual signature. And in particular, they express a lot of genes that are traditionally linked with tissue repair. That is really an unusual feature for response against a pathogen. So this led John to test the possibility that these cells were able to control and limit and control tissue repair. And Sanjay in the laboratory was able to visualize the association of those CD8 T cells with the skin in the context of tissue injury. There is here a wound biopsy that is generated in the mice, and you can actually see via confocal then the CD8 T cells generated against this bacteria can actually accumulate at the edge of the wound, really showing that there may be really an intriguing tropism of cells generated against the microbiota and the injury site. And this is a very interesting site because, of course, it's a site of high vulnerability in which you can be exposed to pathogens, so they may enhance an immunity to pathogen. But at the same time, this actually led to the possibility that these cells may be important for tissue repair. And I'm just going to summarize the finding of John. What he was able to show, indeed, that when the cells were generated, they were indeed able to promote the tissue repair of the animal. And this is visualized here by looking at the epidermal tongue that is actually a proliferative response of the keratinocyte that measure the healing process that can be enhanced when Staph epi is present. And if the animal cannot generate the CD8 T cells, it's actually a molecule that is important for the generation, this response is no longer seen. So this actually supports the idea that this immunity to a commensal is actually very different than immunity generated to a pathogen, and not only is able to boost immunity, but at the same time is able to actually promote fundamental tissue physiological responses. So in summary, the microbiota can control the immune system via many, many, many different mechanisms. Some of them will be direct, and this will be the ability of the microbiota to enhance the cytokine production in the tissue that broadly can act on lymphocyte, allowing these cells now to produce cytokine and have effector function. So very non-specific enhancement of the tonic activation of the tissue. But there's, of course, way more complex phenomenon. And although these microbes are not invading, they are clearly constantly sensed and probed by the immune system. There is clearly the generation of cognate response to this microbe, and we actually have shown this in the antigen-specific responses. This process is really distinct from the one you generate to pathogen because there is no inflammation. But the CD8 T cells that go in the tissue have the capacity to enhance antimicrobial defenses, but importantly have also a flexible ability of also promote tissue repair. And this actually tells us that exploring immunity to the microbiota may really help us to understand really the association within this microbe and the immune system, but also the physiology of the tissue and how immunity to a microbe is really tightly entwined with the physiology of those sites and is really able to just have complex function on the tissue. So to come back to the point, I don't know where the field is going, but I think we have much work to do to try to understand how each microbe is really able to communicate with the immune system. But really, by trying to understand the physiological response to those non-invaders, I think we can better understand how the tissue are wired to sense microbiota, to try to understand and mimic this phenomenon if we want to boost immunity, and at the same time utilizing molecules or antigen produced by these microbes as a way to actually promote immunity locally. And I think there is a lot of research that could, over the next few years, when we kind of understand the antigen and mechanism that can lead to novel way of tackling tissue responses and inflammation. So I have to thank all the people that have done the work. Of course, I have shown some of the work that was done by Shruti that have actually started her group recently. I will show some of the work of John Linham that is actually was a wonderful postdoc that unfortunately is going to leave this week. Don't leave. I think you're around there. Anyway, also great work by Julie Segre that has actually collaborated with us over the last few years. Jason Branchley has actually provided us with some of the resources to explore in primates responses. Great collaboration with Michael Fishback to looking at the mechanism. Ongoing collaboration with Barbara and Stefan, who actually is going to present a work, some fascinating work after me, and many other people over the last few years. Thank you very much for your attention. So, yeah.
That was wonderful. We have one question. Just ask your question into a microphone.
What can you tell us about the messengers of the beneficial effects of some of these organisms to the T cells or to other organisms?
The messenger, the mechanism, you mean?
The messenger, the chemicals that give the clue.
Oh, in terms of the specific response in this case?
For a T cell response or for any other protective effects.
So there's not going to be one mechanism, and that's what I'm trying to explain. In some cases, it's going to be canonical feature. For example, in the context of Corynebacterium, it's going to be a structural element in the cell envelope that is important, macrolic acid.
Sure.
In other cases, it's going to be soluble mediators that come from the metabolism of the microbiota that can go to the bone marrow and affect hematopoiesis. In other cases, the sheer production of TLR ligands that they also express. I mean, they share the same molecules and pathogens. So the bottom line is actually they utilize a whole arsenal of molecules and factors to constantly tickle the immune system in a way that actually allows it to respond in an appropriate manner. So there will not be one mechanism. There is actually a multitude of mechanisms that are employed, some of them soluble, some of them linked to the microbes, some of them product metabolism of the microbiota, and so forth.
What about the vast abundance of short-chain fatty acids that they're talking about?
That's definitely one of the important mediators of the relationship with the immune system, because of course short-chain fatty acids can intersect not only with the generation of regulatory cells, but also of epithelial cells, biology, and so forth. So short-chain fatty acid is one of the product of the metabolism of microbiota that can of course have a profound effect on the immune system.
So Yasmine, the CD8 response at the edge of a wound, is that a general response in other tissues than skin?
Remain to be assessed.
Huh?
So I think remain to be assessed. I think one of the limitations of the field at the moment is the number of antigens that have been tracked is extremely low. Therefore, there is no many tools to track them. So, for example, a group that Danlitam has actually generated sums. We are generating sums. I think we need, as a community, to generate way more tools to track the complexity of these responses, to be able to visualize them via tetramer stainings and generating of tools to track them in vivo. So, remains to be assessed in other tissue.
Any other questions? Don't be Arthur.
I'll ask two simple ones. How many different types of organisms, or whatever you call them, in the microbiome?
Different types? Even like different groups of microbes?
Yeah, I mean, is it in the millions or tens of thousands?
Oh, it's supposed to be, I think the estimations is a broad estimation. There used to be 10 to the 14, it has actually now become 10 to the 13 recently, the GI tract. Or you speak about the number of species we have.
The number of species.
So for bacteria, it could be around 1,000 per person, but it's probably not really well done. And for other microbes, clearly has not yet been done really in a way that is actually appropriate. So at any given time, there will be, if you look at the skin, the GI tract, another maybe around 1,000 bacterial species, but it's probably an underestimation.
Okay, and then the second question is, okay, when I wash my hands to get rid of the pathogens, am I really hurting some of the microbiome too?
It really depends. If you're a doctor, I hope you can wash your hands. But if I will say that actually, of course, this is something that is important. The flu season, all of us have to wash our hands. But the reality is actually our skin is really colonized by a really physiological microbiota. When we actually just wash, we don't really change. Those microbes are really embedded in deep structures. Nonetheless, harsh treatment that can be utilized may potentially alter the relationship with these microbes. So I think you can wash your hands, and if you're a doctor, please do.
You mentioned in passing the use of fecal transplants.
Yeah.
What's the current status of that?
So there is actually, the current status is at many different levels. So of course, the first line has been transplant of microbiota, and actually there is different companies that actually just, now give microbiota transplant. But of course, the next step is to create consortiums that will be actually well controlled, because I think the next generation of therapy linked to the microbiota will be to have a very controlled way of delivering different class of microbes. So people actually are now exploring the association within different microbes in the context of Crohn's disease, for example. There is actually ongoing studies trying to link that to autism and other phenomena. So this is really something that is in development. But the next generation of therapy will be likely to create a very defined consortium that can actually be given instead of actually just microbiota transfer. So.
So I'm just curious about the slide you showed where there was a certain clade of, I think it was Staphylococcus that produced a specific protein response in mice. And so since it was a specific clade, were you able to identify any genetic components that were specific to that clade, mutations or accessory genes?
Yeah, so this is really in development. So we actually have an ongoing collaboration, the group of Michael Fischbach and Erin Chen in his laboratory. And she's actually made a lot of different mutations in the region that is different, that is relatively small. And there is actually evidence that actually deletion of one region can actually affect the behavior. So this is actually in development, but I think using this comparative genetic approach can actually help us in some cases understanding really the mechanism by which these microbes are able to engage the immune system. So, yeah.
Is it part of evaluation of new drugs now to test, when the FDA has to test some new drug, is part of it now?
So your point is an important one, because it has actually been shown, and indeed the metabolism of a drug can actually be affected by the microbiota. I don't really think this is really embedded in clinical trial yet, but I think it will be probably something very important to move forward to be able to actually stratify people based on responders or non-responders, or the product of a drug, or the half-life of a drug, and the kind of microbes that are present. I don't think this is something that is part of the classical routine of clinical trial, but I really hope this will be something that will be part of the future. Yeah.
And no other questions? Yes, here.
I was just wondering if you and your lab have done any work with the B cells response.
B cell responses?
In this context.
So we didn't, but actually it has been done by others to look at the antibody responses and actually the survival of B cells. Yes, this is actually, other people have done it. We didn't, but yeah.
Okay, well, there'll be time.
Thank you very much.
I think, Yasmine, thank you very much.
Thank you, Vin. It's a pleasure to be here and to talk about the microbiome. I'm a physician scientist and have studied for a very long time translational immunology in humans, and come to mouse work from that direction, as I will show you. So the phenotype of all mammals, from mice to non-human primates to humans, is really driven by the metagenome. That's a textbook knowledge fact at the present. And this metagenome is the combination of all of the host genes, and that is outnumbered by the billions of genes from the microbiome. So together, the host gene and the microbiome drive most of the diseases that we know, and more studies in humans now perform microbiome analysis in the complex diseases that we see in our patients. But this metagenome should not be seen isolated. So when we study mice, for example, mice are not in a sterile environment. They live in the natural world as we think humans also live. We live in a complex environment. And mice and humans and all mammals adapt to that environment. So in the natural world, in the case of mice, mice are exposed to a variety of inflammatory stimuli that come from pathogens, viruses, worms, all kinds of diseases, and also toxins, mold that they're exposed to. And we would argue that the constant exposure to these inflammatory stimuli have some advantages to the host. They drive a symbiotic relationship that a microbiome is basically selected to confer some survival advantages to the host. which we show here as an example as a kind of protective green shield. And in contrast, in our animal models that we use to study complex diseases in humans, we are basically creating an artificial laboratory world. We are housing these animals in isolators under very controlled and restrictive conditions. And we are re-deriving them in a germ-free status repeatedly, and then basically separating them from their naturally co-evolved microbiome. They also have a microbiome, but that has been selected in the laboratory world. So as a basis for this presentation, we would argue that there's a laboratory meta-organism, which we use for most of our experiments, and there's a natural meta-organism. And this influences the kinds of results that we get from animal studies, because we would argue that the laboratory mechanism, a meta-organism may have lost this protective shield that is created by some symbiotic host microbiome interactions. So therefore, we arrive at the hypothesis that laboratory mice lack host microbiome interactions that are physiologically important and found in the natural world. And we would like to ask the question, can we learn anything from a naturally co-evolved microbiome? So coming from the hepatitis C immunology field, we know that by studying a persistent virus that has co-evolved with a host and basically persists in the host for decades, for 30, 40, 50 years without killing the host, has developed a very intricate knowledge of the host immune response and has known many more molecules that scientists don't know yet, and has developed escape strategies. So by studying hepatitis C, we have learned about intracellular pattern recognition receptors and many other molecules that were previously unknown. So by studying a naturally co-evolved microbiome, what can that teach us? So this is the basis, and this is the type of question that a postdoctoral fellow in my lab, Stefan Rossart, a physician scientist from Freiburg in Germany who joined about 4 years ago, asked when he started his postdoctoral fellowship. And he said all of these questions are common sense. Yes, maybe they are common sense, but they are very difficult to address in a research setting. So he basically incorporated a very unique approach, and he developed a way to address these questions. And this really speaks to the intramural research program at NIH as well, because it created a model which has infinite possibilities for further studies. So Stefan basically said that in the laboratory, we are comparing in microbiome studies germ-free mice and antibiotic-treated mice with lab mice that were raised in barrier facilities. But what could we learn if we really compare them to a wild mouse in a natural environment, to Musculus domesticus? And how can we study these? So how can we find wild mice that we can study for, in our case, immune responses? So wild mice originate from Northern Europe and from Japan, and were brought in with ships to the US East Coast many, many centuries ago. And they were really the basis for the current standard lab strains in the laboratory. And so Stefan went out and he said, I'm going to trap mice. And he went out into the natural habitat of these mice. So wild mice live in close proximity to humans, and in cold environment, in the fall, in the winter, they move closer. They live in horse barns or close to houses or human settlements. So he went out to 8 different locations that he selected in Maryland. This is a map here. Bethesda is at the bottom. You see all of these colored dots. These are basically horse barns without pest control at a sufficient distance from NIH so that we would not encounter any research facility escape mice there. And he trapped more than 800 mice, which was very, very difficult and a really challenging endeavor. And to make sure that these mice were really not in any way escaped laboratory mice, we performed a very complex genetic analysis. The overarching aim for this was also to find genetic relative of standard laboratory mouse that is closely related. So basically the closest wild living kin of the standard laboratory mice, in our case, standard Black 6 mice that we could find. We collaborated with geneticists at University of North Carolina, and that was Fernando Pardo Manuel de Silva. And he had a 28,000 SNP array, which basically allows the identification of mice at the subspecies level. And you see here the different mice strains that he had collected already from different countries in Europe, shown in green, in Asia and other continents. And you see that the Maryland mice, we show only 4 of them here because all of the dots would be overlapping, are really clustering in the clade of Musculus domesticus from northern European countries. You see the country codes next to the individual dots and the mice from Maryland as the filled green dots at the bottom. And standard laboratory strains, such as Black 6 from the 3 main vendors that we use here, and all the other ones are shown in blue, basically in very close genetic proximity. Mus musculus castaneus and Mus musculus musculus are more distant genetically. So if you perform a formal admixture analysis with complex statistics, then you see that our Maryland mice are really among the mice that are closest related to the standard laboratory strain Black 6. They come up on top in this graph. And the further the dots are on the right, the more shared ancestry is there. But there's really no statistical significance, I would say, between the top 5 or 6 of these mice. But it's nice to see that the Maryland mice are just on top, so they're very closely related to the standard laboratory stain Black 6. So what about their microbiome if they live in the wild? So at the microbiome level, and this is the gut ileocecal microbiome, there is a very great difference between the wild living mice and the Black 6 strains from different vendors, shown in blue. And the wild mice from locations A, B, and C basically show an overlapping cluster. Stefan Rossard also sampled the wild mice in the fall of several subsequent years, and he showed that in each fall season, basically there was overlap of the microbiome. So there seemed to be a stable Maryland microbiome out there that was very distinct from what we see in Black 6 mice from the standard vendors. So when you characterize this at the phylum level, you see the differences. Each bar represents one mouse, and you see the wild mice on the left and the laboratory Black 6 mice on the right. And there were many more firmicutes in blue in the lab mice than in the wild mice, and there were more proteobacteria and bacteroidetes in the wild mice. And at the phylum level, there were statistically significant differences, both for high abundance phyla and at the bottom right for low abundance phyla. And also there were differences at the level of order. So what do we do with this data? How can we use this for research? So this was the next experimentally very challenging task. But Stefan developed methods to biobank viable microbiota from these mice, from the luminal and tissue-adherent ileocecum. And he selected donor mice based on their adult status. They shouldn't be juvenile mice. He categorized them. He developed scores. And he also tested all of the donor mice of the microbiome for common pathogens that would not be allowed in our animal facility. So you see here the results, serological and PCR testing of wild mice from locations A, B, and C for the common bacteria, viruses, and meiofauna. And to our surprise, we found quite a great percentage of wild mice that tested negative for all of the pathogens by both serology and PCR that are not allowed in the animal facility. And so this allowed us then to select donors and to basically transfer the biobank natural gut microbiota from the wild mouse donors to pregnant germ-free Black 6 mice, where they colonized the mice And these mice were pregnant, so they transferred via the natural way the microbiota to their offspring. And this resulted in a colony of black 6 mice that were colonized with a wild mouse microbiota, here shown in the middle as labeled as wild R, so recipients of the wild mouse microbiome. And their microbiome was very similar to that we had identified in the wild mice. And as a technical control, we also transferred black 6 lab mouse microbiome to germ-free black 6 mice, just to make sure that the transfer didn't change anything. And these mice are shown on the right side as labeled as lab R, so recipients of the lab mouse microbiome. We then performed machine learning, and Brian Vassallo, a student in the lab who came after college and is now a graduate student at MIT in Boston, used indicator species analysis to basically identify operational taxonomic units that are indicative for the lab mouse microbiome and those for the wild mouse microbiome. And he showed here that these indicative OTUs remain stable and detectable over all of the 4 generations of mice that we followed and characterized. So this then created a chimeric meta-organism, the offspring of these pregnant female mice that had received the wild mouse microbiome via 3 oral gavages. They were naturally colonized. They combined the best scenarios from both worlds. They had the tractable genetics of standard black 6 laboratory mice, and they also contained fitness-promoting microbes from the wild mouse microbiome. So we therefore hypothesized that they would have this protective shield that we think has developed by evolution in nature, and that they would show a different response to common disease challenges than the standard laboratory mice. And of note, since we had excluded all of the pathogens, we basically excluded the influence of any acute or chronic infection. So really, we had only the homeostatic microbiota that Dr. Belkaid referred to in her talk. So we then asked the question, in our disease models, do natural microbiota promote host fitness of these genetically lab mice? And we used a virus challenge model and then a toxin and mutagen model. So for the virus challenge model, we challenged the mice with influenza virus. So these were the offspring of the pregnant mice that had to receive the wild mouse gut microbiome. And we showed that these recipients of the wild mouse microbiome, here shown in green, basically survived an infectious dose of influenza that was lethal for the standard lab mice, and also for lab mice that had received the lab mouse microbiome. And this was seen for both female and male mice. And the difference was apparent very quickly. So if you just measure weight loss, you see that already 2 days, 3 days after infection, the mice with the wild mouse microbiome do quite well and have even a small increase in weight, whereas the other groups, the blue groups, immediately lose weight. And this was associated with a better control of virus titer early after infection in the mice that had received the wild mouse gut microbiome. So why is this the case? We performed lung histology and saw much less bronchiolitis and alveolitis in the wild mice, in the mice that had received the wild mouse microbiome, as you can see by more aerated space at the bottom slide in the lungs. And this was associated with a decreased cytokine and chemokine protein response in the lung tissue. You see all of the green bars represent the mice that had received the wild mouse microbiome. And for each of these chemokines, cytokines, except for IL-10, which is an immunosuppressive cytokine, an anti-inflammatory cytokine, they had lower levels. So we would think that the wild mouse gut microbiome confers traits that abrogate excessive inflammation in this mouse model. And we know that death in humans due to influenza infection is not due to the pathogenicity of the virus, but due to the host response and to an excessive inflammatory response. So then we used our second model. We wanted to use a completely different model that is also relevant for humans. And we used the model of colorectal tumorigenesis. So in this model, mice are given 3 periods of 1 week of dextran sodium sulfate in drinking water, which basically creates inflammation in the colon. And then they receive at the very beginning of the observation period an intraperitoneal injection of a mutagen AOM. And with this treatment, you see immediately a difference in inflammation score. The mice that had received the wild mouse gut microbiome had less inflammation than the mice with the lab mouse black 6 microbiome. And when you count the number of tumors here visualized by red dots in the slide on the left, you see for both the number of tumors and the tumor area in the colon significantly lower scores for the mice with the wild mouse gut microbiome. So again, it's a reduced response with reduced inflammation in this model. And since we had such a long observation period, we saw something that usually is not observed in these models. We saw that there was also protection from colorectal cancer. So here at the bottom in the histology, you see a Mobert stain, which basically stains mucin and adenocarcinoma. And this was only observed in the lab mice and in the mice that had received the lab mouse microbiome, but not in any of the mice that had received the wild mouse microbiome. And again, if you also give an invasiveness score, then again, the mice with the wild mouse gut microbiome do much better. So to summarize this, I can summarize here. So in the natural world and in the laboratory world, you have both a specific meta-organism, but we would argue that in the natural world, there's a natural meta-organism that is driven by, for example, diverse inflammatory stimuli. And in the laboratory world, there's a more restrictive environment. There's also a microbiome, but the meta-organism is not exposed to these environmental challenges that you would otherwise see in nature. We had hypothesized that natural microbiota beneficially modulate host responses to inflammatory stimuli and created a model where the gut microbiome can be biobanked and transferred into germ-free mice, which creates a chimeric meta-organism with tractable genetics from the lab mouse and the microbiome of the wild mice. And here we see increased host fitness and limited inflammation in lethal influenza virus infection and colitis-associated tumorigenesis. So we think that this model, which we are now using to evaluate protective mechanism in a vast number of diseases, that this may enable the discovery of protective mechanisms that are relevant in complex diseases in the natural world. As physician scientists, we also hope that maybe these mice may model better complex diseases or model vaccines or other responses that are difficult to translate from standard laboratory mice even into non-human primates and later into humans. So with this comes the acknowledgement slide. So as I mentioned in the presentation, this work was really developed and executed by Stefan Rossart, a postdoctoral fellow in the laboratory. So really one person driving this research, and Brian Vassallo was a postbac student who stayed for 2 years and helped and is now a graduate student in Boston. Ashley Hunter is a new postdoctoral fellow who basically has replaced Brian. And then this was possible due to the unique environment of the NIH. We had collaborators at NIAID, John Udell's group that helped with the influenza virus infections. We've collaborated with Yasmine Belkaid. Kazuyo Takeda from FDA performed the histological analysis. And microbiome analysis was done initially at Baylor College of Medicine and then at the NCI here on campus. And the genetic analysis from Andrew Morgan and Fernando Pardo Manuel de Silva really added all of the unique genetics that I showed in these slides. Thank you very much for your attention.
Thank you, Barbara, for bringing this whole new dimension to
In your model, you transfer essentially the gut microbiome, not the microbiome, let's say, of the lungs or the skin, and the effects of the gut microbiome to the model of influenza infection. Have you specified how it works? At what level you have? I mean, yes, you showed lower inflammatory response, but is the microbiome of the lungs changed?
We have not analyzed this, so we have deliberately transferred the gut microbiome. We are now looking at other body sites and other mouse colonies, but in this study we have not done this.
So the animals with the wild microbiome, do they live longer, and are there any behavioral changes?
There are behavioral changes, which we are now following up on. We don't know whether they live longer because we basically use them for experiments.
Other questions? Please go to the microphone and we'll.
Infections in mice, classing laboratory mice. So I was wondering if you've tried things like Coxsackie virus and enteric viral infections that have been traditionally hard to infect in the standard laboratory mice, given like Julie Pfeiffer's work.
Not yet. We've been working with other collaborators at NIH, Alan Scher's group, Dragana Jankovic, on Toxoplasma gondii, for example, and also see health benefits there.
Thank you. It's a very interesting talk. So I have a question which is speculative on your hypothesis. So would you think it would be a big leap to take from rats to the populations in developing countries where they do not have so much of protected shields of environments of growing, when their houses or air conditioning or anything, but they are a lot more exposed to natural environment, which can be hostile or not hostile. So do you think that human populations in developing countries could have microbiomes within them that are better to contend with infectious diseases? Do you think you can extrapolate this hypothesis all the way there?
This is a really interesting question. So a lot of studies are actually going out and studying native populations, studying, for example, hunter-gatherers in Africa, studying their microbiome, which is very different from the microbiome that here in Maryland prevails. And if you just think about the hygiene hypothesis, of course, the microbiome has huge effects on asthma, on Crohn's disease, on all of these diseases that are common in Western countries and not so much common in countries where there's more exposure to microbes and also infections.
To what extent does a newborn baby have the microbiome of an adult? Does it have most of the microbiome, or does it have to acquire it over a period of time? Or do we know?
But we know that a lot of the immune response is basically transferred from the mother, antibodies, but we also know that the microbiome changes during early life. I have not studied that specifically. I don't know.
I think it also depends whether the child is born by cesarean section or delivery, and that's become a big thing.
Yeah.
The difference in
basically the main conclusion is don't be afraid of all microbes. Not of all, not in all situations.
So if I understand correctly, you've been able to maintain the wild, natural microbiome from the mouse. You can sustain that in the laboratory in a form that you can now introduce it into all kinds of experimental models. Is that correct?
This is exactly
so is that
case.
the direction that your work is taking, to see whether there are differences in the immune response?
Yes, correct. And what are the mechanisms, and how does the baseline immune response already differ in these mice from that of laboratory mice? The studies in humans that I did before We know that even exposure to basically sub-infectious doses of some viruses, just because of a high exposure rate, influences the immune response and influences the immune response to later actual infection. So this is one of the references that are included. So we know that this has relevance for humans as well. The question that we always get, which doesn't come up now, is whether we humans are more like the lab mice or more like the wild
Some
mice.
are and some aren't.
We would hope that it's more the situation in the natural world.
Well, while other questions are brewing, let me ask another one. The literature is filled with studies of the microbiome in virtually every kind of disease you can think of, but let's talk for a moment about gastrointestinal and liver diseases. People look at the microbiome before and after transplant, before and after this drug or that drug, and so forth. In your general gestalt about this, I realize that what is your view as to, quite frankly, the value of those studies? Do they advance our knowledge of how disease is interacting with the microbiome, which is then interacting with the host? Or how would you go about studying that kind of a problem? Is this, in your opinion, perhaps not the best way to do it?
This comes back to the title of this whole seminar today. Where does the field stand? I think we just don't know enough yet. A lot of the human studies are still collection of stool samples and just characterizing the microbiome in different populations, in different stages of diseases, after different interventions. And this all has to be seen in the context, not just of the disease, but also of the entire history of a patient and other kinds of exposures or diseases or infections or antibiotic treatment that these patients had. So it's very difficult to interpret. It's not just asking who is there in terms of microbiome, but also what is the function of this microbiome. Maybe different microbiota or different communities of microbiota, even though they're different, they trigger the same functions. We don't know that yet. So at the moment, it's still very descriptive and a lot of cataloging, which I think is necessary.
So I have a question about maintaining of the microbiome in the lab mouse, because so the diet in a mouse in the wild is very different than the lab mouse. So what effect do you think the diet in the lab has on the microbiome? And then another question, what behavioral changes did you see?
So with the diet, we were very surprised. So all of these changes in social structure and living environment, day-night cycle, temperature, and most of all diet, did not affect the maintenance of the microbiome in this colony that we kept for 4 to 7 generations. So we were very surprised by that. We did not give them the natural diet. We gave them the standard lab mouse diet. We think now, we have done further studies, that this microbiome is very stable per se. So there may be less drift in terms of environmental challenges. But we know also from studies that are published on wild mice that are trapped in different seasons, that then with changes in diet, there are big differences in the microbiome. Just in each same season basically circulates back, so it's resilient. It comes back to the same that it was a year before in the same season. Yeah, the other question, I'm sorry, I can't answer yet. I hope maybe in the next demystifying seminar.
What do you think would have happened if you would have given those mice that had the very stable microbiota across generations antibiotics and then fed them the kind of bare bones laboratory diet? Do you think that would have changed, or do you think they would have eventually?
We don't know. That's one thing that could be tested, or other types of environmental challenges to see how far, how large is the resilience? Does it come back to what it was before or not? Maybe it's also less well adapted to some other kinds of challenges that are not encountered in nature out there.
Well, we started this with a reference way back to Metchnikov, so maybe we should end this with a question dealing with Metchnikov in the modern sense. So when people go to the drugstore, or maybe it's the supermarket, I don't know, and they buy something that's called probiotic, how alive are those organisms? Do they do any good, really? Have they ever been tested? My understanding is they're not regulated by the FDA. They're regulated by the Department of Agriculture as a food supplement. So do we really, honestly, scientifically know anything about the probiotic
Yeah, I would
aspect?
be very hesitant answering this question, but I know from the hepatology practice and literature that any type of supplement that is not regulated by FDA, that can be just taken over the counter, that's supposed to be good for your health, can have very detrimental effects on liver, on gut, on anything. Difficult.
Well, I always thought that if you really wanted to follow on the lactobacillus idea, giving it by mouth made no sense whatsoever. You really should give it in some form by rectum, which is not exactly a commercially palatable approach, but probably stands a greater chance of doing something, maybe. At any rate, we want to thank you very, very much, Barbara.
Thank you very much.